Hello children, I am going to take the lesson 10 Microbes in Human Welfare, and today's topic is the sewage treatment plant, which is coming at the latter part of your lesson, but I am taking this in my first video. See, this is an aerial view of the sewage treatment plant. Okay, there's lots of water, wastewater generated from households, from the cities and towns. This municipal wastewater is known as sewage. It sometimes may contain human excreta also, but now we know the human excreta goes separately to a septic tank, separate tank. But the water that is coming from the households, like say from the bathrooms, from the kitchen, from the garden, etc. All these, there's another outlet. Where are these water going to? Which water bodies? Can it be dumped into the water bodies like streams and rivers directly? No, it cannot. So first it should be treated and made less polluting and dumped into the water bodies. But it is after this treatment it cannot be suitable for consumption. It is made less polluting and then dumped back into the water bodies. So in today's topic we are going to see how the sewage water that is generated from the cities and towns are being treated in a sewage treatment plant. See, here I have indicated some households. So this indicates a city or town. The water coming from the kitchen or the bathroom, etc. are taken for treatment. Now the first part of the sewage treatment plant. This is a sewage treatment plant. The first part of the sewage treatment plant is a tank which deals with physical treatment, and this treatment is known as the primary treatment. So first it undergoes primary treatment or physical treatment. Okay, the primary treatment or physical treatment, the water that is collected is from the households are being collected in a very huge tank, and this tank is known as the sedimentation tank. Okay, now what happens in this physical treatment? There are two stages for this treatment. Okay, one stage is sequential filtration and the other step is sedimentation. So in the first step what is done is the floating debris are removed. So in the sequential filtration the floating debris are removed. The debris are removed. What are the debris? The debris include plastic bottles, plastic packets, straw, leaves, sticks, etc. All that are floating. That is removed sequentially by filtration. Okay, so I am removing. So all this is indicated is removed. Then the remaining solid like matter like pebbles, stones, soil which forms the grit is removed by sedimentation. So here we have the floating debris removed by sequential filtration and the grit which involves pebbles, stones, soil removed by sedimentation. Now I want you to make a comparison with the centrifugation tube. When you centrifuge or allow a compound to settle, say if I mix tea powder with water, I am making a combination of tea powder with water in a test tube, okay, or a muddy solution, that is mud and water, and you are allowing it to settle. What happens after some time? After one day, if you keep it for sedimentation, what happens? You can see the upper part, you can see it becomes a very clear solution, and this upper clear solution is called the supernatant is called the effluent. When you compare it to a tank, it's called the effluent, but in a centrifuge tube, you called it as supernatant. So when you say it in context to a test tube, it is called supernatant. This part is the supernatant. Okay, this clear solution is the supernatant, and the settled sediments are called the sediments. Okay, when we compare it to a test tube, But in a tank, this clear solution on top will be called effluent and the settled sediments will be called sludge. So remember the two terms. The two terms we are going to use further when I am dealing with one tank with the another tank. The two terms I'll be coming across is effluent and the second term sludge. And I made a comparison with a centrifuge tube. After centrifugation or after sedimentation, the supernatant, the clear solution on top, we call it supernatant when in context to a test tube, and the settled sediments, we call it as sediments. In the case of tank, we don't say supernatant and sediment, we use that upper clear solution after sedimentation as effluent, and what settles at the bottom of the tank as sludge. So with these two terms I am going ahead. Okay, so now after the first treatment, which is called primary treatment or physical treatment, what happens? I told you the first step is sequential filtration, where the debris is removed, like all the floating debris is removed sequentially, and the second stage is sedimentation. In the same tank, it is allowed to settle. The sediments that settles form the sludge. Now this sludge specifically of this sedimentation tank is known as the primary sludge. Okay, and the sludge is removed from this tank. Okay, and this sludge is used as filling for foundations for buildings. When you make buildings, you dig the earth and you make a foundation for the building, and you pile it to make a very strong foundation. To fill that, as a filler, what is used is primary sludge. So it is not going a waste. This sludge is dumped to make the strong foundation of buildings. Clear? So that is the first treatment, primary treatment or physical treatment. It involves two stages. Sequential filtration to remove the floating debris. The debris includes leaves, green foliage, plastic packets, plastic bottles, straw, etc. And sedimentation is, it settles in the very same tank, it settles. What and all settles? The soil particles, pebbles, dirt, etc. settles. That is called the grit. It settles by itself, and this forms, see, dark colored clayey substance at the base of the tank. You must have seen this, and this is called the sludge. I told you the two parts which we are going to always refer to. This upper part is called the effluent, which is the supernatant, and the lower portion, the settled sediment, is known as a sludge. Now this sludge is called the primary sludge, which is used as filling for making buildings in construction sites. Now this is the primary treatment, it is over. Now we are going to the secondary treatment. Secondary treatment is also known as biological treatment. Here it involves three tanks. See, first tank, second tank and the third tank. The first tank is called the aeration tank, the second tank is called the sedimentation tank, and the third tank is known as the anaerobic sludge digester. So once again, after the primary treatment or the physical treatment, we are moving on to the next treatment, that is the biological treatment. The biological treatment involves three tanks. The first tank is called the aeration tank, second tank is the sedimentation tank, and the third is the anaerobic sludge digester. In the aeration tank, what happens? As the name suggests, you can guess, aerobic bacteria is present. So very important, aerobic bacteria is present. You know the difference between aerobic and anaerobic bacteria. Aerobic bacteria, they decompose the organic matter in the presence of oxygen. Now, this aerobic bacteria is facilitated to grow vigorously into flocks. So the conditions that are prevailing in the aeration tank is conducive for the aerobic bacteria to grow vigorously into flocks. What are flocks? Flocks are aerobic bacteria entangled with fungal filaments. We know fungus, we have learned the structure of fungus. It has filaments called the hypha and the mycelium. So the entangled mass, this mass of threads should be entangled with the bacteria. That entangled mass of fungal filaments with bacteria is known as flocks. I hope you have understood. If I scribble here, see, if the white is the fungus threads or hypha or the fungal filaments, the bacteria will indicate with another color, say green color. These two constitute a flock. So what are flocks? Aerobic bacteria entangled with the fungal filaments. See, these white threads are the fungal filaments, and the green indicates the bacteria. So when these aerobic bacteria associated with the fungal filaments, it form flocks. So here the conditions that are prevailing in the first tank called the aeration tank is to promote the vigorous growth of aerobic bacteria into fungal or into flocks. Clear? Now to create this condition, what is done? Air is pumped. So what are the requisites? Air should be pumped. Air is pumped, and not only that, it is constantly mechanically agitated. Agitated in the lab, and how we do agitation? By stirring. Here also there is some stirring so that it gets aerated. So remember the 3 A's in this aerobic in this aeration tank. The first A stands for the aerobic bacteria. Aerobic bacteria is present, and the aerobic bacteria vigorously grows into flocks, and the conditions to allow this growth is air is pumped. So the next A is the air. Air is pumped deliberately, and not only that, there is also constant mechanical agitation. Okay, and under these circumstances, what happened? The aerobic bacteria becomes into flocks, and I told you what are flocks. Aerobic bacteria entangled or associated with the fungal filaments. So this is maintained. How long this water is held here? It is held here until the BOD is brought down significantly. So this is maintained till the BOD is brought down low. Now later I will explain what is this BOD. So the water is held in this tank till the BOD is significantly reduced. So once it is reduced, see, the water goes to the next tank. See, now where did I give the outlet? The outlet is at the effluent. See, I drew the pipe at the effluent, not at the base. If the pipe is drawn here, means sludge. The pipe is drawn on top, means the effluent. Now the effluent is made to pass to the second tank called the sedimentation tank. As the name suggests, sedimentation tank is meant for sedimentation. Now here what settles here? The flocks. Flocks are heavy. The flock settles at the base. So if I am showing pink color as the flock settles at the base. We know what is flock, the entangled mass. It settles at the base. So there are lots of flocks. They settle at the base, and then there is a supernatant which is called the effluent. Now here did you see one outlet here? Now this, again we have a supernatant called effluent, and this settled base is known as the sludge. This sludge is called activated sludge. Now this sludge is called activated sludge. The use of this activated sludge is, it serves as an inoculum for aeration tank. So in the aeration tank we have to constantly feed it with aerobic bacteria. So aerobic bacteria should be always be given here. Aerobic bacteria should be supplied constantly. How it is supplied? This flock which is settled at the bottom, which forms the sludge. The second sludge here is the activated sludge. It is taken from here and it is placed and it is added here. So the sludge is added here. It is serves. So this term used here is inoculum. Now here I want to make a comparison with a real life situation. How your mother makes curd. You know how your mother makes curd. What she does is, into fresh milk which is boiled and cooled, what she add a pinch of previous curd. The curd which she had the previous day or previous week, that a small pinch or a small spoon of the previous curd is used. That contains a lot of lactobacillus, and then she puts it in the milk and she covers it overnight, not in refrigerated. She puts it out with room temperature, and you can see next day you get curd formed. Okay, so what was the previous curd serving as? It served as an inoculum. So same way here, this sludge which is taken from the sedimentation tank is used as an inoculum here. That is to provide aerobic bacteria. I told you this aeration tank is a previous tank. It requires aerobic bacteria. Aerobic bacteria need not be produced in the lab or from the research scientists. The aerobic bacteria can be constantly supplied from the sedimentation tank, because in the sedimentation tank what settles at the base is the flock, and the flock is very rich in this aerobic bacteria. So a bit of this aerobic bacteria, small portion of this aerobic bacteria or sludge, and therefore the sludge is known as activated sludge, is taken and transferred here to supply the aerobic bacteria. So you understood the function of the sedimentation tank. As the name suggests, it allows sedimentation. Here what happens, it is left undisturbed. For sedimentation, what is important is it should be undisturbed. What settles here is the floc. The flocs all settle. Now once the floc all settle, next see what comes. What is the next connection? It's not effluent. I didn't draw a pipe from the effluent. The sludge is made to the remaining part of, so only a small portion of the sludge is used as an inoculant, very small portion. The remaining major part of the sludge is taken to the third tank. The third tank is called the anaerobic sludge digester. As the name suggests, which bacteria should be present here? The anaerobic bacteria. And this anaerobic bacteria digests the sludge. The action here to digest the sludge which comes from the previous tank. It digests the sludge. So the name, it gets its name. So it's very easy. You can understand what happens in the last tank from the name of the tank, anaerobic sludge digester. So the sludge that is taken from the previous tank is dumped into this tank, and anaerobic bacteria digests this sludge anaerobically, and the end product of this digestion is a mixture of gases like hydrogen sulfide, methane and carbon dioxide. What are these gases? These gases are biogas. They are highly inflammable and can be used as biogas. So in the anaerobic sludge digester, anaerobic bacteria is present. It digests the floc. What is the floc? It digests the bacteria as well as the fungus, producing byproducts like hydrogen sulfide, methane and carbon dioxide, which can be used as a biogas. Now look at the pipe that goes out. It's going from the effluent, the remaining effluent. See, now the pipe outlet is from the effluent part, the remaining effluent, where it is going, see where it's going, it is dumped into the water bodies. Now it is dumped into the water bodies. See. Now after treatment, it has reached the water body after making it less polluted. So once again, let's analyze or once again take a recap of the biological treatment. Biological treatment involves 3 stages or 3 tanks. The first tank is the aeration tank, second tank is the sedimentation tank, and the third tank is the anaerobic sludge digester. After the primary treatment, the effluent is dumped into the aeration tank. As the name suggests, remember the 3 A's. In this tank, there is the aerobic bacteria, and the aerobic bacteria grows vigorously into flocs. The conditions required for this growth is air. Air should be provided. So air is deliberately pumped, and not only that, there is constant mechanical agitation. Under these conditions, what happens is a lot of aerobic bacteria turns into flocs. And I told you what are flocs? Aerobic bacteria entangled with fungal filaments. Now this water is held here until how long? Until the BOD is brought down significantly. The biological oxygen demand is brought down very low. Once it is brought down significantly, the water after this is made to pass to the next tank called the sedimentation tank. Now you have to specify which part. Is it the sludge or the effluent? The effluent. See, the arrow comes from here. The effluent is taken to the next tank called the sedimentation tank. As the name suggests, it is allowed to sediment. The water after the aerobic tank is treatment is made to pass through the sedimentation tank. Here the water is allowed to settle. Now what settles here? The floc settles. Because it's heavy, it settles at the bottom. Now this settled portion is known as sludge. I told you this sludge is known as the activated sludge. It serves as an inoculum to provide aerobic bacteria to aeration tank. The remaining sludge is passed to the, that's why the outlet came from the base, is passed to the third tank called the anaerobic sludge digester. Here anaerobic bacteria is present which digests the bacteria and fungus of the floc to produce a mixture of gases like hydrogen sulfide, methane and carbon dioxide, which are used as biogas. The remaining portion are the effluent. This effluent is made to dump, or it is dumped into the water bodies. Now it has been made less polluted. Okay, so here we have seen how the heterotrophic bacteria is used in sewage treatment plant to make the water less polluting. Clear? Now BOD is an abbreviation. BOD is biological oxygen demand. It refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. So, once again, what does BOD stands for? It refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. The purpose of the bacteria in the water is to utilize the organic matter. In the process of utilizing the organic matter, what is taken up by the bacteria? Oxygen is taken up. So, it is also a measure of the uptake of oxygen by the microbes, and also an indication if there is a lot of organic matter. See, there is a lot of organic matter in the tank, it means the BOD is high, which means it indicates high pollution. Okay, so you understood the relationship. Organic matter, if the amount of organic matter is high, it indicates that the BOD is also high, and this is an indicator that the water is highly polluted. Clear? So, the purpose of the bacteria is to bring down the organic matter by utilizing a lot of oxygen, which means that low BOD indicates the amount of organic matter is less. That also is an indicator that the level of pollution is low. So, indirectly BOD is a measure of pollution. So, if you get any water sample and the water sample shows high BOD, you must understand that the water is highly polluted. If it is having low BOD, that the water is less polluted. I hope you understood what is BOD. It can be referred first as the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. That is a technical definition. It is also a measure of the uptake of oxygen by the microbes. It indirectly also measures the level of pollution. High BOD means a lot of organic matter is present in the water, which means high pollution, and the vice versa is the case of less pollution. Clear? Now, sewage treatment plant action plans taken place that has taken place in India is the Ganga action plan and the Yamuna action plan. Ganga action plan and the Yamuna action plan. Both plans were initiated by the Department of Environment and Forestry, okay, to bring down the pollution of the Ganga and the Yamuna, very important rivers of India. And though it was, see if I show you this cutout, it was actually initiated by late Prime Minister Rajiv Gandhi. The objective of the Ganga action plan was to reduce pollution from agricultural runoff, human defecation, cattle wallowing, throwing of unburnt and half-burnt bodies into the river. That was the objective of the Ganga action plan. But though it was initiated, to some extent it was not implemented well. But now the present Prime Minister very actively under the Swachh Bharat mission has brought about this Mission Ganga. Okay. He is very famous for Swachh Bharat, and he has come out with this Clean Ganga project. Okay. And he has vowed, the Prime Minister Narendra Modi had vowed during his campaign in Varanasi that he would bring back the lost glory of Ganga by initiating policies and programs to improve the water of the Ganga. Okay. So, this is the latest, and it is an ongoing process, very active process. And the plan is brought into action, though it is initiated long time back. So, once again a quick recap of the sewage treatment plant. I told you it involves 2 stages or 2 treatments, primary treatment and secondary treatment. Primary treatment is physical treatment. It just involves 2 stages: sequential filtration to remove the floating debris, and sedimentation to remove the grit. After the physical treatment of primary treatment, next is the biological treatment, which involves bacteria. First is the aeration tank, second sedimentation tank, third is anaerobic sludge digester. The aeration tank, what happens there? Aerobic bacteria is introduced, mechanical agitation is done, and air is pumped. This facilitates the aerobic bacteria to vigorously grow into flocks. This is maintained until the BOD comes very low. Once the BOD is brought down, the water, the effluent is transferred to the sedimentation tank. The sedimentation tank, what happens is the flock settles at the bottom. and very small portion of this flock or very small portion of the sludge, which is called as the activated sludge, is used as an inoculum for the first tank called the aeration tank, that is to supply aerobic bacteria. The remaining major portion of this sludge is made to pass the third tank called the anaerobic sludge digester. I just told you a clue to remember what happened. Anaerobic sludge digester, that is anaerobic bacteria digest the sludge. What does the sludge contains here? Bacteria and fungus to produce a mixture of gases. The gases are hydrogen sulfide, methane and carbon dioxide, which is used as a biogas, and the remaining portion, that is the effluent, is made to pass into the water bodies or dumped into the water bodies. So now it is made less polluted, and I told you the two action plans, the Ganga action plan and the Yamuna action plan, was initiated by the Ministry of Forest and Environment to bring down the pollution levels of both the Ganga and the Yamuna, mainly from agricultural runoff, from human defecation, cattle bellowing, then from the dumping of dead bodies or ashes into the river. Okay, so I hope you have understood this topic, sewage treatment plant. It's very important from 5 marks as well as 3 marks point of view. Thank you.